Today we will be I am Dr. Swathika, Assistant Professor Biochemistry. What is the central dogma of molecular biology? Before going to the translation proper, we will be reading about what is the central dogma of molecular biology. It means that the message from the DNA has been transferred onto RNA by way of transcription. And from DNA to DNA, the message can be transferred by way of replication. After transcription, the message has been transcribed onto RNA. And from the RNA, the message will be transferred into the protein by way of mode called translation. And we finally get the ultimate required or desired protein to bring about the various cellular effects which are desired. So what is translation? It is the process of translating the nucleotide sequence of an mRNA or messenger RNA into the sequence of amino acids of the corresponding specific protein. The information which helps or aids in translation is the genetic code. Here you can see the messenger RNA, which is running in 5' to 3' direction, to which so many bases, nucleotide bases are attached. A stands for adenine, G guanine, cytosine, and uracil. Cytosine and uracil. Instead of uracil in DNA, you have thymine. That has been replaced by uracil in RNA. So RNA, again RNA similar to DNA, is read. The message is read in a triplet genetic code. So here the 3 bases are being read. So if you take the example of here, CUA, that is cytosine, uracil, and adenosine has been read, and the complementary to this basis has been found in the video of the amino acid surrounding, and the complementary bases are being paired to the bases which are present in the mRNA. So G is being 1 is paired with cytosine with 3 hydrogen bonds, adenine with uracil with 2 hydrogen bonds, and uracil with adenine by 2 hydrogen bonds in the direction of 3' to 5'. So this is the tRNA to which the incoming amino acid which has got sequences complementary to the bases present in the mRNA is attached. And the cRNA will aid in delivering the amino acids to the mRNA and helps in synthesis. This is how proteins is being synthesized with the codes present in the RNA, and we regulate amino acids, and the respective amino acids are synthesized, resulting in formation of polypeptides. The incoming tRNA carrying the amino acid which has got to which the coding is complementary to the coding present in the next sequence of RNA. Next to C. Translation ideally commences from 5' end of mRNA, that is from the amino terminal. So mRNA is basically read in 5' to 3' direction. In eukaryotes, translation mostly occurs in nuclease. So what are the components required for translation? We have 5 major components. One is the mRNA or messenger RNA. Second is the tRNA, which carries the corresponding amino acid. Third one is the ribosomes. Ribosomes has got subunits. So 70S ribosome in case of prokaryotes play a role in translation, and 80S ribosome plays a role in translation in case of eukaryotes. And other ribosomal proteins like rRNAs. And fourth factor is the protein factors, which are helpful in translation, which include eukaryotic initiation factor, eukaryotic elongation factor, and RF restriction factor. Fourth one is the energy source, ATP and GTP. So here you can see this is the mRNA, just got a cap, I' cap, adenosine cap, and polyadenylene thymine, the 3' region. After the untranslated region present in the 3' end. Here also you have got untranslated region in the 5' end, And the first codon which initiates is called as the starting codon, which is encoded by A. So this is the coding region in the mRNA. So to which the subunits of ribosomes will come and associate to form the wholesome ribosome, and this will come and bind to different coding regions in the mRNA and initiate synthesis of simultaneous synthesis of polypeptide chain, which is called amino acids. Prokaryotic ribosome has got two subunits, 50S subunit and 30S subunit, to constitute the 70S ribosome. 50S subunit is made up of 23S ribosomal RNA, 5S ribosomal RNA, and 35 proteins. 30S subunit is made up of 16S ribosomal RNA and 21 proteins. These subunits will associate to form the 70S ribosome. Eukaryotic ribosome is made up of 60S subunit and 40S subunit, which will constitute together to form the 80S ribosome. 60S subunit is made up of 28S rRNA, ribosomal RNA, 5S rRNA, 5.8S rRNA, and 14 proteins. And 40S subunit is made up of 18S rRNA and 33 proteins. S is nothing but spread bucket, is a measurement unit. This two will associate to form the 80S ribosomes. So this is the tRNA adapter molecule. It has got 4 stems. One is the acceptor stem. Second one is the D loop. Third one is the anticodon loop. Fourth one is the variable loop. And sixth one is the TC loop. So this acceptor loop has got an amino acid sequence of TCA at its 3' end and a hydroxyl group attached to the last codon at the 3' end, which is the accepting arm for the incoming amino acids to be associated to the polypeptide chain. This is the D loop and this is the anticodon loop. Here in the end has got sequences which is complementary to the bases present in the mRNA, to which helps in decoding the information present in the DNA. So this will go and bind to this anticodon loop will go and bind to the mRNA. So what are the steps in translation? So these are basically the most important or an outline of steps in translation. Activation of amino acids, initiation, elongation, termination and release, folding and post-translational modification. So how do amino acids get activated? So an amino acid will be activated by the enzyme amino acid tRNA synthetase. That is, activation means by phosphorylation of amino acid. So a molecule of phosphate group will be attached to the incoming amino acid, making it activated with the help of enzyme amino acid tRNA synthetase. So ultimately we get an amino acid tRNA. The incoming amino acid after getting phosphorylated will be attached to the tRNA, that is forming amino acid tRNA. This is the activation of amino acid. The second phase of translation after activation of amino acids includes initiation, elongation, and termination. Initiation can be divided into 4 steps. First, ribosomal dissociation. First, before going and getting attached to the mRNA, ribosomes has to get dissociate itself. So if in case you take for the eukaryotes, 80S ribosome will get dissociated into its subunits 60S and 40S. And then happens the formation of 43S pre-initiation complex. Third one is formation of 48S initiation complex. And fourth one is the formation of 80S initiation complex. So here you can see dissociation of 80S ribosome. So here 80S ribosome is dissociated into 60S and 40S by way of certain restriction factors or whatever, which helps in dissociation of the ribosomal sub. Second one is the ternary complex formation. So the first codon, which is the AUG codon or the start codon, will encode for the first amino acid methionine, and along with that, in eukaryotic initiation factor and also the dissociated subunit 40S, which has got attached to the 3 and 1A factors, along with this methionine and this eukaryotic initiation factor 2C will get attached and form a ternary complex, which is nothing but 43S pre-initiation complex. So here you can see activation of mRNA. So this is how formation of 43S pre-initiation complex. Third step is formation of 48S initiation complex. So once this 43S pre-initiation complex is formed, to which one more thing, mRNA with its associated factors A and 4B and 4F attached to the cap of mRNA will come and get associated itself to the 40S subunit of ATS ribosome along with other factors which aid in translation and resulting in formation of 48S initiation complex. After formation of initiation complex, 48S initiation complex, we have another step where there is formation of ATS initiation complex. Here you can see the mRNA, the starting codon, the amino acid which encodes for the starting codon as methionine, along with a part of the ribosomal subunit and along with the factors. After this, what happens? 60S subunit, which is dissociated at the beginning, will come and associate itself to the 40S subunit to form the 80S initiation complex. Rest of the things remains the same. So what are the difference in initiation between the eukaryotes and prokaryotes? In eukaryotes, while binding of mRNA to the 40S subunit, 5' end of cap will bind to the ternary complex, and mRNA is scanned and it is positioned in such a way that the incoming amino acid, the methionine, will be present at the P site of the ATS complex. And in prokaryotes, mRNA is not capped. Shine Dalgarno sequence upstream of the initiating AUG will bind to the complementary sequence of the 18S rRNA of the subunit of ribosomal RNA. This is the prokaryotes. The first amino acid in the eukaryote is methionine, as in case of prokaryotes, it is formyl methionine. And the initiation factors involved in translation or initiation of translation in eukaryotes includes 12 or more, whereas only 3 initiation factors play a role in translation in case of prokaryotes. And the ribosomal subunits are 40S and 60S constituting 80S ribosomal RNA, and 30S in case of eukaryotes, and in case of prokaryotes, 30S and 50S together constituting 70S in case of prokaryotes. Second step in translation is elongation. It is a cyclic process, that is, one amino acid will be added at one time, and it has 3 major steps. First step is binding of the amino acid tRNA, that is activated amino acid tRNA to the A site of the ATS initiation complex, and then what happens? There will be a peptide bond formation with the initiating amino acid methionine present in the P site, and with the incoming amino acid tRNA at the A site. And then what happens? This amino acid present in the A site will get translocated to the P site after peptide bond formation, resulting in a dipeptide. So here you can see ATS subunit in the has got P site and associated with the mRNA has got 2 sites, P site and A site. A site is where the coming tRNA with its anti-codon complementary to the codon present in the mRNA will come and bind with the coming amino acid attached to its acceptor step. So this is the initiating codon AUG, to which our tRNA with the initiating amino acid methionine has already come and attached. So once this comes and attached, what happens? There will be a peptide formation between the This sequence CCG will code for the amino acid proline. So hence, after it coming unattached, then there will be a peptide formation to methionine. And after this happens, there will be a translocation of this entire proline from this tRNA, charged tRNA. We can say that accurate tRNA is charged tRNA onto the methionine, and this tRNA will be held free and it will move to the E site to get released. So here again you can see A site, B site, and E site. So peptide tRNA with the first yarn amino acid will come and attach to the B site, and then the incoming tRNA will come and attach to the E site, peptide formation, and the translocation of the amino acid N-1 to the B site. So this tRNA then moves to the E site, and this sequence of amino acids again will attach to the tRNA present in the B site, which got shifted from the A site, and that is how peptide bond formation and translocation of tRNA, uncharged tRNA, will happen. And then from E site, the uncharged tRNA will get released. So during this process, we need energy source, which is nothing but GTP. So here, this is the step 1, which is binding of amino acid tRNA to the A site. Here A site, N+1 amino acid is coming, charged tRNA is coming and binding with the anticodon on the tRNA and codon on the mRNA. Second step is peptide bond formation. That is, the whole sequence of amino acids present in the tRNA in the B site gets forms bond with the income already bound amino acid present in the A site, and then this will get shifted to this, and here, so hence the tRNA present in the B site is free of amino acid. Then translocation. So now you can see this, the newly synthesized polypeptide with everything is present in the tRNA in the A site. Now what happens? This entire molecule will get translocated from the A site to the B site. And this A site is now free to receive the incoming amino acid present in the charged tRNA. So for the entire process at each step, we need GTP. Energetics. Equivalent of 4 high energy bonds. The charging of tRNA molecule with the amino acid moiety requires is the hydrolysis of an ATP to an AMP. So 2 phosphate molecules are attached, and the entry of amino acid tRNA into the A site results in hydrolysis of 1 GTP to GDP, and translocation of the newly formed peptidyl tRNA in the A site into the B site results in hydrolysis of GTP to GDP. So in the first step, during charging of tRNA molecule, 2 phosphate groups, and the second and third step, 1 phosphate group is being utilized. So totally 4 phosphate groups has been utilized. And the third final step of translation is termination. Termination happens when there is a stop codon appearing in the A site. So no tRNA with an anticodon will be available for recognizing the termination signal. So the releasing factor F1 will recognize the stop codon in A site. Here you can see the animation. I'll play it again. So here there is a what to say. First, there is a P site tRNA to which there is attached amino acid, and there is translocation of after peptide bond formation, translocation of this amino acid tRNA to the P site. And here UGA is one of the stop codon, and there is no tRNA available to recognize it. So what happens? This release factor will come and bind to the stop codon and cause dissociation of all the ribosomes and replaces this polypeptide chain from the mRNA. And now the mRNA is free. This is how termination of translation occurs. So here you can see again, this is the stop codon, and the B side, the entire polypeptide chain is attached to the tRNA, which is present. And to the stop codon, there is no tRNA recognition because no tRNA is available which can recognize the stop codon. And now here comes into the picture the releasing factor as well as the energy, VTP, will come and bind, and this will cause dissociation of the subunits of ribosomes and also dissociation of the nucleosynthesized polypeptide chain from the tRNA, and hence we get the free polypeptide chain, free tRNA, free ribosomal subunits 40 S and 60 S, and other factors which favor in termination of translation and the mRNA. So polysomes, what are polysomes are multiple ribosomes which are present in the same mRNA molecule form a polyribosome. Hence a single mRNA molecule is copied into a big protein molecule. So here you can see mRNA to which so many ribosomes gets associated and results in formation of polypeptide synthesis. This will synthesize the desired polypeptide of this region, and here it will synthesize the polypeptide of this region, and similarly here it will synthesize the polypeptide of this region. Regulation of translation. So most genes are regulated at the level of transcription itself, but some are regulated at the level of initiation of translation, and others are by altering the stability of mRNA. So regulation of rate of translation. Here the initiation is being regulated by regulation of eukaryotic initiation factor 4E, which controls the rate of initiation. So initiation factor 4E and 4E binding protein. So when insulin is acting upon this will cause kinase activation and release of and phosphorylation of 4E binding protein and release of 4E binding protein from the eukaryotic initiation factor and 4E. This eukaryotic initiation factor 4E also got phosphorylated, to which the eukaryotic initiation factor 4G will come and bind. And after this, there is an incoming of eukaryotic initiation factor 4A, which will all together form the eukaryotic initiation factor 4F complex. This 4F complex is required for attachment to the mRNA cap. This is how it regulates. So when there is no phosphorylation happening, this complex cannot form and which cannot go on bind with the cap of mRNA. So here you can see how ferritin synthesis is regulated at the translation level. So this is the ion response element binding protein, this orange color, and this is the ion response element, this elongation or imagination in the 5' end of mRNA. So whenever there is low iron, we don't want iron to be stored as ferritin. We want the iron to be utilized. So what happens? Whenever there is low iron, there is no translation of ferritin protein happening. Hence, we can't store iron as ferritin. Whenever there is high iron in the circulation, this iron will go and bind with iron response element binding protein and will cause free iron response element present in the mRNA end, enabling translation of ferritin protein. So viruses and translation. So viruses replicate using host cell protein synthesizing machinery, which includes encephalomyocarditis virus, reovirus, vesicular stomatitis virus, and polio and picorna virus, which disrupt the function of 4F complex. So already we have seen how 4F complex will regulate the initiation of translation. So here polio virus will cause dissociation of 4E from 4G, and there is no formation of 4F complex. So this 4E and 4G will go and bind to iron response element. So he separated the unimolecular 4G. If it goes and binds, it will favor translation. Whereas there is no binding, there is no translation happening. Stability of mRNA. How stability of mRNA will influence the translation. So mRNA has got one more entity. There is one more entity called microRNA, which degrades the target mRNA or blocks translation of target mRNA. So mRNA of eukaryotes is relatively stable, which is brought about by poly-A tailing, and the second one is it binds to proteins that prevents its degradation. Antibiotics and translation. How do antibiotics affect translation? Tetracyclines will prevent binding of amino acid tRNA to the A site, hence translation could not happen. Chloramphenicol will bind 23 years ribosomal RNA and inhibits its peptidyl transferase activity. Diphtheria toxin will catalyze ADP ribosylation of elongation factor 2 and inactivates it, and ricin will inactivate 28 years ribosomal RNA. Erythromycin and clindamycin will bind bacterial 50S ribosomal subunit. Streptomycin will bind bacterial 30S ribosomal subunit. Cycloheximide will inhibit peptidyl transferase activity in 60S ribosomal subunit. And next, last part is protein folding and post-translational processing. So once the protein polypeptide chain has been synthesized, it has to be folded into a biologically active form. So to achieve or assume its native conformation with the formation of appropriate hydrogen bonds, van der Waals forces, and ionic and hydrophobic interactions to form a functional protein. So in this way, the linear mRNA is converted into a three-dimensional structure of a protein. Some newly made proteins do not attain their finally biologically active conformation. They have been altered by some of the processing reactions before that, called as post-translational modification. So amino and carboxy terminal modifications. So again, methionine capping at the fibrine end, and ubiquitin attachment at the carboxy terminal end. These all will determine the fate of the protein. If ubiquitin is attacked, it is subjected to proteolytic cleavage inside this chaperone molecule in the eukaryotes, and there's methionine attachment, fibrine for capping, and loss of signal sequence is one of the post-translational modification happen. So modification of individual amino acids, phosphorylation of serine and threonine can happen. Gamma carboxylation and hydroxylation also would happen, which is also come under post-translational modification. And glycosylation, attachment of carbohydrate side chains to the protein, can also happen. Either it can be mannose groups or galactose group or muraminic acid, N-acetyl glucosamine or N-acetyl galactosamine. And proteolytic processing, like in case of zymogens, it has to get undergo proteolytic cleavage to produce the active protein to produce its desired action, which happens in case of digestive enzymes, clotting factors, where zymogen or inactive precursor is converted to the active protein. And addition of prosthetic groups, addition of isoprenyl groups, formation of disulfide cross-links, as in case of glutathione and also in insulin. So these are the post-translation modifications which happen after translation of protein before it gets converted into a native biologically functional protein. And with this, translation is over. Thank you.